to deliver a talk which is basically focused on the circular proteins as a molecular scaffold for highly functional, because I choose this particular word because we always think about how we can stabilize the any proteins or enzyme which will have the more applications and more durability when we go into the industrial scales. So with that, I would like to start my talk. So as I mentioned here, circular proteins. So let's first understand what exactly these circular proteins are. So as you can see in my slide here, that what we know usually as a linear protein, we know as a biotechnologist that there is a protein which is a building blocks, right? Which is a amino acid which is a building blocks of protein, and proteins are major part in our daily life routines. So what we know, the protein which is having an NC amide terminal or carboxylic terminal, as you can see over here, which are basically connected with the peptide bond, very known bond, peptide bond, as a like a covalent bond, right? So circular protein is itself is a unique class of biomolecules, and this is characterized by their head to tail closed loop structures, which have even emerged as a very important player in modern biotechnology, specifically for number of field like industries, number of industries. So you can see the linear protein versus cyclic proteins. So why we are calling this as a cyclic proteins? Because they are had to tell connected via peptide bond or covalent bond. This can be done synthetically also, or in already this is available in the nature. So we are talking about cyclic proteins. So let's say why we are too much interested in why we are talking about the cyclic proteins. Well, their architectures has been imbued with the exceptional stability and resistant to proteolytic degradation and higher thermal stability, having acid or alkali resistant, and also it is highly stable toward proteases. As we know, proteases can chop the protein. So these specific proteins, which is known as the circular proteins, are highly stable against these things. And also these are the major things. Because of that, we cannot go for the protein based drug designing if it is a linear, because we have to think about stability before delivering into particular systems. Nowadays many anti-cancerous peptides or proteins are also there, but once we try to deliver, it may lose the activity. So we have to think a little bit different, that how we can first stabilize the protein of interest, and then let's go for the designing of protein based drug. So it is stable, and because of that, it broaden an n number of uses, specifically the applications which I'm going to focus over here. We can say the first applications, the cyclic proteins can improve the pharmacokinetic and dynamics of the particular molecules. Like there are several examples, including insulins, very well we know as a biotechnologist, antimicrobial peptides, growth hormones, and peptides or protein based vaccines, which is very common these days. Number two, industries. The major focus on industries, like there are number of enzymes. And as Indians, I also know that we ask students to do their dissertation to isolate the enzyme from particular bacteria or particular fungus, and we don't even think about how after isolation, how we have to carry those things, how we have to maintain those things. The research should not be, in my point of view, the research should not be just stay in isolation, confirmation, and that's all, or purification. We have to think beyond that. So enzymes like amylase, lactase, pectinase, glucose oxidase, and cholesterol oxidase, patase is a very unique enzyme which eat the plastics. It is known as a plastic eating like enzymes, lipases. These are having huge applications, and day-to-day life we are using those enzymes. So we have to think about how we can stabilize those kind of industrial enzymes to enhance their uses. Next application is environmental applications, like biophysical properties, for example wastewater treatment. Now I'll give one example of enzymes which is well known as a horseradish peroxidase enzymes. It has huge applications in wastewater treatment, and also like degradation of phenolic compound to non-toxic phenolic compounds, bioremediation, also sequestration of heavy metals. Another applications like diagnostics. In diagnostics we are using antibodies or enzymes, so we have to think about the stability again there. So immunodetection, glucose oxidase biosensors, biochemical testing kits, even the recent one COVID detection kits, those are all enzymes, right, and antibodies. So let's talk why these naturally occurring cyclic peptides or proteins are that much having superiority. So because this nature has been already adopted this configuration, and there are like molecules which protect their host cells from the microorganism attack. Because example, antimicrobial peptides are the key components of innate defense system, and these antimicrobial peptide has been first identified as a circular peptides, as a circular peptides which was highly stable, and it act as a defensing molecule in the system. So because of this kind of superiority, the nature has been already adopted this system to protect themselves. So these cyclic peptides are highly resistant toward several pathogenic microorganisms, and for that we thought of to work on this a little bit more on this protein cyclization or peptide cyclizations. So the question comes here, how we can make the cyclic proteins or peptides. Basically it's engineering things, or there are having several number of methodology. For example, a long back, decade back, the scientists started with the chemical methods, but chemical method was suffered with the size of the molecules. Like we cannot go more than 18 to 20 amino acids long, and as well as the method itself was highly costing because of the chemicals which use here, protonation and deprotonation chemicals, which is very expensive. So for higher molecular weight peptides or protein, this method is not feasible method. Then next come to the biological method. The biological methods are having advantage over, I must say, chemical methods. I will give some examples like sortase mediated cyclization, butylase mediated cyclizations, or even genetic programming by an unnatural amino acid incorporation during the synthesis of the proteins. So you can say I mentioned biological method because sortase is an enzyme, butylase is again one enzyme, and we can go for the genetic code reprogramming directly. There is one more method which is known as intine mediated cyclizations. Basically intines is considered as a embedded useless molecules, I must say, which has no functional in the genome. But later on it was found that intins or introns are not just a embedded genetic elements, but it having its own function. It has the auto splicing functions, which is unique functions which do not need any enzymatic reaction. So unlike the other biological method for protein engineering or cyclization, this intine mediated protein cyclizations does not have any burden on the cells. As also we can embed those molecules into the downstream or upstream of our target to achieve the things. But only the problem with this system is this system, as you can see, once the splicing happen, it leave the target protein hydrolyzed form also, or half of the proteins, or 75% proteins also go for the cyclic proteins. So this method is unique because we can have no worried of size limitations. Also, we can have high scales production and cost effective method, and this can be done in vitro as well as in vivo. So the intin molecules, as we are talking about intin mediated cyclization over here. So the intin molecules, as I mentioned, is a genetic elements, and now there are several intins has been already identified, and that found that the intins are the very unique molecules which we can use for the protein engineering, and number of applications are also now coming out. So these intins are basically having splicing, as I mentioned, splicing applications which do not require any of the enzymatic reactions. And the applications, if we talk about applications of intin molecules, we can use itself this intin molecules as a protein labeling molecules or protein cyclization molecules for protein engineering, and for gene therapy also, for protein purification also, as well as for the synthesis of biosensors. So here we started this intin molecules for our research for the cyclization. So we choose the GFP. GFP is a green fluorescent protein as a model protein. So this is the linear protein, as you can see, where we say the structure, the three-dimensional structures with perfect beta sheet, helicals, and loop. And as I mentioned, the linear proteins or linear peptides or linear product which will have N and C terminal opened, right? And that is why they are very flexible. And because of the flexibility of the linear product, they are having less stability, and they can easily lose their conformational integrity, and they will be like inactive. You must have gone through the research article that says particular enzymes having optimum temperature of only 25 degrees Celsius, or particular pH like 5 or 6. So because of, if a little minor pH or little minor temperature shift will be there, the variation will be there. Because of the open structures, more flexibility, these proteins can lose their stability and activity as well as. And once the activity is lost, we know that there is no use of any purified product. If we are having in our hand, it is not used at all. So let's see how the cyclic GFP look like. So cyclic GFP, if we are going to engineer, it should be closed, completely closed by one loop to another loop via the covalent bond or polypeptide bond as well. So the resulting molecules will be highly rigid. It will maintain the rigidity, and the entropy will be maintained, as well as the conformational rigidity will be there. Therefore they are more stable. And of course, a peptidase, if I'm saying it is highly stable toward the peptidase, we know that the function of peptidase, it starts chopping from the terminal. So there is no terminal, hence the peptidase action will be completely gone. So cyclic proteins are peptide. Now the major question comes here, that how we are going to identify the cyclic product. Like other conventional linear protein, the best way to detect the cyclic protein via LCMS Malditov analysis, right? And it need purified cyclic proteins, and of course this method is expensive method, as we know that. So any techniques which used to identify the cyclic product from the linear counterpart, as I mentioned, some part of the product go for the hydrolysis also. So of course, in the mixture, you will have cyclic as well as the linear molecules also. So we have to identify, and the only one method which is very well known is the Malditov analysis. Now the question comes, if all time we are going to do Maldi analysis, we have to think about cost effective manners also, because we can't just to go every time Maldi analysis. It is expensive analysis, right? So the thing is how to detect, how to identify with cyclic out of the linear one. And of course, the major question here is the purification, because we have to pick the cyclic product out of the linear mixture. So here the question comes, how we are going to do. So for that, we engineered the tag itself, the tag which is known as a CMYC tag, and this CMYC epitope we did the engineering. So this CMYC tag, as you can see over here, we engineered this tag, and we know that there are some specific amino acids which are needed if we are going to work with the intin mediated cyclization. So intin mediated cyclization, there should be certain property like that has ability to use both N and C terminal, as well as the amino acid which play very crucial role with the this intin mediated cyclization is the serine, threonine, and cysteine amino acids, which are the very important and very essential amino acids. So with that, we pick this MIC tag, and the idea was very simple. This MIC tag is a very simple, 11 amino acid no, the small tag, and we thought of engineered this C tag, CMYC tag, and we have to find the very crucial junction to split this tag, so that the half part of the tag will be at the N terminal of the linear protein, and half part of the tag will be at the C terminal of protein. And when the intin will assist for the cyclization, this tag should come into the closure, and it should make a proper peptide bond, so that this linear loop will be connected via peptide bond to one by another. And the idea was, if this tag will be connected with each other, then only the anti-MYC antibody will be bind to the target molecule. If it is a split, and if the product is let's say hydrolyzed product, then this tag will be splitted out, so antibody will not going to find any antigenic molecule on the target linear product, and antibody is obviously not going to bind with the linear product. So specifically, if complete a tag sequence will be there, then only your antibody is going to bind, and in the Western we will get the signal. So this was the idea which we did the engineering. So we applied the same strategy, and how we did, we split the tag, and we splitted the tag, and this X is nothing but any three amino acid, as I mentioned over here, the cysteine, serine, or threonine. The target would be your protein, like here we put GFP as a target, and this is the half splitted part of the MIC tag. Then we will have intin molecule over here, and another intin molecule at the N terminal, and how it will be going to do the splicing. So it will exactly do how it is getting spliced out in the system, in the genome system. So same chemistry it will apply, and finally over the N to S acyl shift, the linear, if it is a 100% cyclization or 100% acyl shift is happened, then 100% there is a chance of cyclic product synthesis. Otherwise, some part of the product will also go for the hydrolysis if it is not perfectly undergo for the NON acyl shift process. So likewise, we will have both. The chance of the product would be 50%, maybe sometime 50, as I mentioned, 50, 60 or 70%, both product will be there. And from that, we have to pick our target. As I mentioned, we develop a tag which will only and only specifically detect the cyclic product only. So here we confirm that the tag which we have engineered, we went for the several round of mutation and shifting of the serine, cysteine into different position, and the perfect position which gave the higher specificity in the Western blot, we picked that one. So in our case, we found 8 position of the MIC tag with cysteine amino acid was the best position, and we were having higher detection of our product. So these are the different positions, as you can see, 6 position, 7, 8 position, 9 position. And at the 8 position here, we got Western blot result, very high specificity. So we fixed here and we picked this mutation, and we split it right from the glutamic acid and cysteine molecule, like that. And we cloned this part with N and C terminal of our target. Now, how the selective purification will work for the cyclic protein? So we develop an idea along with our construct. We also design in such a way that later we can go for the immobilization of this and a protein or antibody, or even now commercial bead is available. That commercial bead is known as anti-semic agarose resin. So for that we purchased this antimic antibody, which was conjugated in a agarose bead. And the idea was very simple. As I said, if it is selectively specific in the Western blot and only picking the cyclic product, then it is in the purification strategy also, the antibody which is conjugated over here will only and only going to bind or interact with the full length of the product, cyclic product, not with the linear. So after the binding, we can go for the elimination by washing the product. So the linear will be eliminated out, and this cyclic product can be eluted out as a final product, as a pure product. And then further we can go for the whatever the choice of applications we are going to see. So likewise, we successfully demonstrated this strategy, and we also demonstrated successfully the purification strategy over here. We did the purification and the only cyclic product. As you can see, the upper band is the linear, the lower band is the cyclic product. We picked out the lower band, and then we apply the same strategy with the another peptide also, because GFP was as a model protein, but later we also cross check with any other peptide or higher molecular protein. So here we choose, we design some peptides, short peptides also, that is known as the RGT peptide, which we use in the application of other industrial enzymes. So the same chemistry we did with another, and we published this data in analytical biochemistry after the cross verifying how much stable this cyclic protein is over the linear GFP. So you can see the linear GFP lost its activity, but cyclic was the highly stable. In even it retained it color even in a higher thermal or denaturation. Guanidium is a highly harsh chemical, and it is known as a enemy of the protein or enzyme. Even a trace amount of guanidium, if above the 50 millimolar guanidium is there in the buffer, it will quench out, it will kill all the activity of the enzyme or protein. But we tested this against the guanidium hydrochloride to see what is happening, and we concluded that this stability is coming because of the rigidity and cyclic conformation of the GFP. So as I mentioned that the protein which we choose GFP, it was a model protein, but we also wanted to demonstrate this strategy for pharmaceutically important proteins via end-to-end cyclization. And for that we selected horse radish peroxidase. Why horse radish peroxidase? Maybe the question is coming in your mind that why we have particularly selected horse radish peroxidase. Because horse radish peroxidase is having huge applications. It is very well known analytical enzymes which has tremendous applications. That is why it has a huge demand. But the major drawback of this enzyme is that it is isolated from the plant, that is known as the horse radish peroxidase plant, and scientifically it's known as a Armoraceae rusticana. And this plant is having more than, now it has been recently found that more than 35 isoform. Can you imagine 35 isoform are there? So obviously the downstream purification will be really huge, and it will cost a lot, and it is take a lot of energy as well as the manpower, and of course the main thing we talk about as a scientist, the cost. So it take a lot of cost for downstream processing. So we have to think about any alternative tool, that how we can synthesize this HRP, and how we can deliver into the industry because of the higher demand. So this this HRP, we started with the known uh, strategy, which was very long back, back to 1999. One group from Germany, Professor Granko, he started this work, but he found that this HRP is highly insoluble, and it express highly in an insoluble body, because the reason is, the first thing, it is a from plant source, and for the folding is not happening into the bacterial system, right? And because of the higher applications, anyway we have to find some good way to make it recombinantly. So in our group, I started this work in during my PhD. So we choose this enzyme, and we did as reported, and we confirmed that okay, this enzyme is highly insoluble, and everything is going into inclusion body. And for intin mediated cyclization, we need soluble fraction of the protein. We cannot go with the insoluble, because in during this expression only, this intin molecule will be spliced out, and only the cyclization will take place. But if it is a refolding is problem, and it is inclusion body, this strategy is not going to work. For that, we again hunted for the different kind of fusion tag. There are a number of fusion tag, as a biotechnologist, you guys also might be know that maltose binding, sumo tag, NS tag. There are number of tag, but we were looking for specific tag which should be very stable, as well as the loop should be very close to each other, so that we can bring together this loop for our purpose. So for that, we found that phosphoglycerokinase. This phosphoglycerokinase is a very well enzyme which participate into glycolysis. So this we excised from the E. coli itself, because it is present in E. coli. All the system is having phosphoglycerokinase. So we excised it one, and we clone this PGK right before the HRP. So after cloning, we did the expression, and we found that after adding this phosphoglycerokinase as a fusion tag, as a soluble fusion tag, we found that the almost 99% HRP was converted into a soluble fraction. Refolding is happening, and perfectly it is going into the soluble fraction. And from soluble fraction, we did the purification and activity assay, and we found that the activity is also a very good to go for the our further next step. So the first task here was solved, that how we can solubilize this HRP, which itself is a big achievement at present also, because there are n number of now group which are working on HRP. Basically four groups are there in entire world which is working on NHRP. So now our work is very well recognized, and we are in a very good to go for the next round. So later we wanted to also because I said this HRP is a plant enzyme, so it need glycosylation and post translation modification for the activation, because this having four very essential component for its activity. That is a calcium chloride, because it's a calcium as a ligand, hemine and GSSG to provide the cysteine bond, as well as the this one hemine pocket, because this enzyme is a heme pocket enzyme. So our idea was to express this HRP right from the system in an active form. Now we have soluble form, we wanted to go for the active form. So here we found one gene that is known as a ferrochylate gene. Ferrochylate gene is again present in bacterial system, and this ferrochylate gene is very much essential genes which convert alpha levulinic acid into the hemine. It basically incorporate like hemine into the HRP. The idea was to overexpress or co-express this enzyme, do the hemine biosynthesis inside the system itself, and then incorporate the hemine during the HRP expression itself into the system, because we don't want it to add any heme transporter or hemine, and hemine cannot easily penetrate if we are providing into the media, because we know semi-permeable, but everything cannot go inside the cellular system. That is the uniqueness of the cell walls, right? So we did this one, and we found that in presence of Fc co-expression, we are getting active HRP right from the cellular system. But only the problem was the low yield. Yield is very low, so we cannot go for the industrial demand. We have to think about something else. So this was proof. We proved that the HRP can be solubilized, as well as the heme can be incorporated during the expression itself, but only the problem was low yield. And then we move toward our cyclization thing, where we utilize the inting. So what we did, now we are having like a system that PGK with HRP. So we exactly use the split system which we use for the GFP. Half MIG tag at the N terminal, half MIG tag at the C terminal, and C intin at C terminal, and N intin at the N terminal. We went for the expression, and we found that even during the expression itself, the HRP with PGK is getting cyclized. We confirm with the MIC antibody and his antibody that yes, this double band, you see, as I said, that 50% is going into hydrolysis. So the lower band is the linear byproduct, but upper band is the cyclic product. So this was confirmed in the Western blot that this product is the cyclic product. And then later we did the purification and activity assay and confirm that it is giving a very good activity, as well as the stability was also good. We wanted to also try to make native HRP size, because after we cloned PGK, the size was very big. So big size, we want to reduce the size, and we wanted to make such a strategy that how we can bring the HRP in its own size. Like HRP is 38 kilodalton natively, and what we were synthesizing with PGK was the 71 kilodalton, very huge size. So we engrafted one more strategy where we put one more intin system. The idea was to express the full length of this cassette along with all the set and eliminate the PGK. This SSP DNA B is one more intin, which is a pH dependent intin. So once the expression will be done, in vitro, we can reduce the pH to 6 pH, and this is 6 pH dependent cleavageable intin. Once the lysate, the crude lysate will be bring into the 6 pH, this part will be eliminated out, and we will have only this part. So the idea was this split intin and this split intin will undergo for the splicing. It will connect this MIG tag with another piece of MIG tag, and we will have only the native HRP, which will have even more application and more stability because of the small size. Then at that construct, we also confirm that our strategy is working. And now from 71 kilodalton, we are, as you can see here, this is the big size, 71 kilodalton. So the second strategy was work out very perfectly. And now we are getting 38 kilodalton of the target, and which was confirmed into the MIC antibody also. After that, we went for the stability assay, where we did screening of the stability towards the chemical denaturant, heat stability, as well as different ranges of pH. And what we found that the cyclic product, as you can see over here, this is 3 molar guanidinium chloride. Cyclic product was more stable, even 15 minutes, and this is 4 molar and this is 5 molar. So even 5 molar also up to 5 minutes, the cyclic HRP was more stable than the linear counterpart. Even in heat stability, you can see the cyclic HRP was more stable than the linear one. And then later, whatever the HRP variants we had, like from the commercial, from our PGK HRP, the first construct, which was the soluble HRP, and this was the refolded HRP, that is insoluble HRP, and the cyclic HRP. We concluded that the experiment for this stability of cyclic HRP is more stable than its linear counterpart, as well as even from the commercial HRP. Even you can see the bar over here, it is more stable than the plant produced HRP. So this is validated more and more with the more time exposures, and in all the experiment we concluded that this cyclic HRP is much more stable than the linear as well as from the commercial HRP, and the stability fold was also very high than the linear counterpart. As you can see, even in 5 molar chemical, for 10 to 15 minutes, 5.6 to 2 fold more stable than the linear counterpart. So with that, I will conclude my talk today. So the conclusion is, for the backbone cyclized EGFP and RGD peptide, we used this two product as a model protein, and we successfully applied that peptides, that RGD peptides, which was very stable after the cyclization, as a in-house peptide inhibitor for the enzyme which is known as a MMP1. This MMP1 is having different classes, and this is basically matrix metalloproteinase. And in our skin, the aging come because this matrix metalloproteinase, once it get activated, it give the reverse effect in our skin. So this matrix metalloproteinase is a very sensitive enzyme which can be easily activated if metal ion is coming into contact. Once this will get activated, it will start chopping the collagens, the collagen chains, and once the collagen will be lost out, then we will have aging problem will come. Also, this MMP is one of the enzyme which promote the metastasis during the cancer progression, because if it is active, then it will start chopping all the barriers and collagens. So what will happen? The cancerous cells can easily migrate it out. So that is why we work on one of the cosmetic project where we wanted to identify the peptide which can inhibit this MMP enzymes. So that was later transferred to CJ Chemical, South Korea, for purpose of anti-aging chemical cosmetic. So this was a very big project which we handle, and now commercially it is available. The cream is commercially available as an anti-aging collagen based cream. So the conclusion here I will draw is the cyclic protein.
P, please close your camera. You are just, yes.
Shall I continue, sir?
Yes, madam, please continue.
Thank you, sir. So with that, today I will conclude my work that the cyclic proteins or peptides can be a next generation elusive biomolecules, in the modern biotechnology world. And also the cyclic proteins and peptides enhance the biophysical properties by increasing the entropy and decreasing the enthalpy, which subsequently provide the structural rigidity also. And because of the structural rigidity, the cyclic protein or enzyme will be highly stable. There are few methods have been developed to produce the cyclic proteins, and because of the limitation of size, this could not be able to come into the commercial point of view. Therefore, we developed this intine based strategy to cyclize any protein using heterologous expression, and which can be highly used into the industrial applications. And also subsequently we demonstrated the split tag engineering, and for this detection we also demonstrated the purification of cyclic products, where we can bypass the MLD things and specific costly purification strategy, as well as we actually I didn't include it over here. Later we work on one of the project which was expression of MYC antibody into the bacterial system. So you can find this paper in my Google Scholar that where we develop antibody itself. We engineer the antibody, we synthesize the antibody in a bacterial system, and we compare the activity with the commercial one, because this anti-MYC resin is highly costly. 1 ml resin is cost around 2000 USD. So we wanted to transfer this technology to the industry, or because for bulk purification, if for example, if industry demand is there and if we are selling our product, so we need to think about the secondary purification strategy, and as well as how we can bypass the commercial bead, and how we can replace the commercial bead with the in-house synthesized antibody, which will be immobilized in a bead, and from that we can utilize for the purification. So this data I have not included. I thought it will be very much. So you can, if anyone interested, you can go to my Google Scholar, that Sushma Chauhan, and you can find, and if interested you can read, where we did the antibody engineering, same antibody, and we demonstrated for immobilization, and after immobilization we did the purification. So we have successfully produced the cyclic HRP using our strategy, and also we have shown for the first time that the cyclic HRP with the higher stability, which will have the enhanced biophysical property, and it will be this strategy can be easily exploited in the modern biotechnology field to improve the biophysical property, and also it will provide higher stability, which will be have, let's say, tremendous applications in the various biotechnology sectors. And understanding the this versatility and the structural intricacy of cyclic proteins, it will open a new avenue for the development of obviously next generation biotechnological tool, and also it will be utilized for the therapy. So with that, I will thank you, and I would like to thank National Research Foundation, Korea, for supporting our research. And with that, I would like to thank you, and especially I will express my thanks to Professor Gopalji sir, and Poonam ma'am, and all the students present over here, faculty members and the dignitaries. So thank you so much.